In this lecture series, we are going to be covering in detail the process of transcription and translation. In previous chapters, we have already discussed how is the flow of information from DNA to protein. That is also known as the central dogma of genetic information. Let's now review how we make protein as humans, but also these processes are found in prokaryotic cells. So, a DNA molecule undergoing the process of transcription is able to synthesize an RNA molecule. Notice that the enzyme that catalyzes this reaction is called RNA polymerase. Now, even though we are not going to study in detail, I want to point out that the process of transcription is reversible, and there are some examples in nature, specifically viruses, that are able to turn RNA into DNA by the process of reverse transcription. And again, we are not going to study it in detail in the chapters 11 and 12. Then, once we have an RNA molecule, understand that through the process of translation, ribosomes are going to turn that RNA molecule into protein. One of the things is that to date, the process of translation is not known to be reversible. We haven't found a system in nature that is able to accomplish that. Let's now go through an overview of the process of transcription. There are some general features in the process of transcription. Understand that the process in which we are making RNA copies utilizes DNA as a template, and we already know that that is the definition of transcription. The process of transcription gives rise to all sorts of RNA, including mRNA, tRNA, and rRNA. Remember that these molecules are staying at the level of RNA. The only one essentially that turns into protein is going to be mRNA because it is the template for protein synthesis. But understand that out of all the different structures of RNA that we have discussed so far, these are the ones that are directly related to protein synthesis because they have a role in this process. Now, understand that when RNA is being synthesized, we're going to only utilize one strand, and that's the one that is going to serve as a template for the synthesis of RNA. Now, when it comes to the enzymes, because now we are synthesizing many units, so we're linking together many units of nucleotides, we are going to create this polymer with an enzyme that is called RNA polymerase. Understand that RNA polymerase, similar to what we saw in DNA polymerase, is going to be synthesizing this new RNA strand in the 5' to 3' direction. Different from DNA polymerase, RNA polymerase do not need a primer. When it comes to the nucleotides that are going to be building this new strand of RNA, we have adenosine triphosphate, guanosine triphosphate, uridine triphosphate, and cytidine triphosphate. Because remember that the only base pairs that DNA and RNA are have in common are ATP, GTP, and CTP. Understand that UTP, it is exclusively found in RNA. When it comes to how RNA polymerase identify the segment of DNA that needs to be transcribed, understand that there's a region in the DNA that are called promoters, and that's where transcription gets initiated. Lastly, polymerases are going to stop RNA synthesis when they have reached sequences that are called terminators. And we are going to be exploring in this chapter in further lectures what some of the mechanisms that are utilized in order to terminate the process of transcription. Overall, transcription in prokaryotes is the one that we are going to be studying in detail. As we're going to see later in the presentation, when it comes to transcription in eukaryotes, it is complex. So I will be discussing only a few features of the process, but the one that we will be diving in and studying in detail is going to be the one in prokaryotes. Similarly to what we saw in DNA replication, transcription in prokaryotes is going to have 3 stages. It's going to have an initiation stage or step, it's going to have an elongation step, and it's going to have a termination step. Similarly to what we have seen before in the initiation step, we are going to have different systems that are going to identify the segment of in this case DNA that is going to be transcribed, and at the initiation step we are going to see how the 2 strands are going to separate. During the elongation stage, we're going to see how the enzymes that are involved in the catalysis of this RNA molecule are going to synthesize the RNA molecule. And during the termination stage, as you can see, the system, the machinery is going to reach a terminator sequence, and that's what's going to release the newly synthesized RNA molecule and the enzymes that are involved in the process. We are going to explore each one of these as we go along. Remember, when it comes to the biomolecules that are involved in transcription, remember that not all of the nucleotides that we have discussed already are involved in the synthesis of this RNA molecule. Specifically, the nitrogenous bases that we're going to see are going to be adenine, guanine, cytosine, and uracil. Remember that thymine is not involved in transcription because that is specifically for the process of DNA replication, because we're making more DNA. Remember that in transcription, we are going to be using nucleotides that have 3 phosphate groups. Because every time we are making these polymers, understand that the electrophile must have 3 phosphates at the 5' end, in this case of the ribose sugar. As I mentioned previously, the enzyme that catalyzes the creation of this new RNA molecule, it is called RNA polymerase. And as you can see, the picture or the image that we have on the left, that is specifically how the RNA polymerase looks in E. coli. This is a representation of the structure. You guys do not need to memorize what are the components of the E. coli RNA polymerase. I just wanted to illustrate that this is going to be a large protein that contains many units in order to accomplish this job. So understand that this enzyme is the one that synthesizes RNA. It is DNA dependent because remember that DNA is going to be its templates. Okay, there are many different types of polymerases, and understand that E. coli polymerase is the one that is best studied and is the one that is mostly used in the laboratory. These are holo enzymes because, as you can see, they have multiple subunits, and that's why I wanted to illustrate it with the image that we have on the left side of the slide. When it comes to transcription, understand that when we are starting with, we have a double-stranded DNA molecule. This double-stranded DNA molecule is going to contain a sequence that, when it comes to the strands, one of the strands is going to be called the coding strand. The other sequence is going to be called the template strand. The difference between the coding strand and the template strand is as follows. The coding strand is going to have the same sequence as the RNA transcript, but the thymines are now uracils in the RNA. We are going to explore this when we go through the worksheet. The other sequence is going to be the template strand of DNA. And the template strand of DNA is what's going to direct the synthesis of the RNA. This sequence is going to be complementary to the template strand, okay? And again, this is what's utilized in order to synthesize the RNA. As I mentioned before, when it comes to the RNA transcript, it is synthesized in the 5' to 3' direction. So that means that the polymerase is going to move along this DNA template in the 3' to 5' direction. So what I want to point out with this is that what we're going to be observing later, and I'm just going to make of it a note of it in this little corner. In exams, if I give you double-stranded DNA, to simplify the process, remember, one strand is going to be 5' to 3'. The other strand is going to be 3' to 5'. I'm just going to put them in different colors. The yellow highlighted one and the green highlighted one. For simplistic view, we're going to assume that the strand that is running 5 to 3 is going to be the coding strand, and the one that is running 3 to 5 is going to be the template strand. And again, this is just to simplify the whole system. If you're given double-stranded DNA, assume that the strand that is running 5' to 3' is your coding strand, while the one that is running 3' to 5' is going to be your template strand. One of the things that we must also point out is, how do polymerases know where to start transcribing? And there is a region in the DNA that is called a promoter. And promoter sequences are going to be DNA that signal the start of transcription. And they also even control the frequency of transcription. What I have right here in this image in the bottom, this is going to be different promoters in prokaryotes. And the takeaway from this image is the different regions that are part of promoters that are in prokaryotes. And I'm just going to highlight them in magenta. So there are 3 specific regions that are found always in prokaryotic promoters, and they are the -35 region, the -10 region of Pribnow box, and we also have the transcription start site, or TTS. Now, I want to specify the reason why this value is negative is because it is upstream from the start of transcription. So if it's a negative value, that means that it is up the road from the transcription start site. The negative 35 region specifically, as we're going to see once we discussed the steps specific to transcription of DNA, this is where the sigma factor binds. The negative 10 region, or the Pribnow box, I'm just going to write it up the way, is what facilitates unwinding of the DNA template. And as you can see, this is a AT-rich region. Lastly, the transcription start site is the location where transcription starts, and understand that this is going to be after the negative 35 region and the negative 10 region. Another thing that I want to specify when it comes to this is that, as you can see, we have different genes of different prokaryotes. These are known to be conserved regions because the nucleotides are very similar when comparing the sequences among organisms.